Good afternoon. Welcome to Labcorp and Median joint webinar. We're happy today to bring you the topic on imaging in oncology trials. Can it help to make go or no-go decisions? Imaging biomarker has a potential to identify key metabolic pathways that are either upregulated or downregulated in cancer cells compared to normal cells. In early drug development, they can help to provide valuable information on the dissemination of the drug and estimate whether the drug reached the target, and eventually to determine appropriate clinical benefit. The aim of today's webinar is to discuss modern imaging biomarkers and how they can be used in oncology trials for early response and disease management, eventually to help drug developers to make go or no-go decision appropriately. We have three speakers today: Professor Laure Fournier from APHP, Dr. Ravi Karra from Labcorp, and myself on behalf of Median Technologies. Labcorp is a leading global science company that provides vital information to help doctors, hospitals, pharmaceutical companies, researchers, and patients make clear and confident decisions. So the empowered diagnosis and drug development capacity, Labcorp provides insight and accelerate innovations to improve health and improve life. Median Technologies is a leading AI-based next-generation precision medicine company. We focus on helping conquer cancer and other diseases through routine imaging tests and imaging service for drug development. With our office based in US, Europe, and China, we operate globally to deliver best-in-class end-to-end imaging solutions for your oncology trials. A practical reminder that you may submit your questions during the presentation in the question panel, and we will answer all of them at a roundtable discussion. So now I'm very happy to introduce you our first speaker, Dr. Laure Fournier. Dr. Laure Fournier is a professor radiologist from Hôpital Européen Georges Pompidou, Université de Paris. Laure is specialized in clinical imaging diagnosis and also being a research fellow from University College San Francisco. Professor Laure Fournier is an expert in big data and imaging of tumor using various machine learning tools. Laure was also the executive member of the imaging group of the European Organization of Research and Treatment for Cancer, where she plays a key role in implementing imaging biomarkers in oncology trials. Laure, we have the privilege to have you today to open discussion and give us the introduction talk on how imaging biomarker can be used in clinical trials. Thanks, Laure.
Thank you very much. Thank you for having invited me. I'm very happy to be with you. So my job is to kind of introduce the subject and to discuss how imaging biomarkers can be used in clinical trials. So these are my disclosures, just to say that of course, I'm not going to spend time, that I do work with some industrial partners. Sorry about that. Just let me go back one slide. Imaging biomarkers in clinical trials can have two circumstances. It can either be a clinical trial to validate the imaging biomarker, but that's not what we're going to be talking about today. We're going to be talking about how an imaging biomarker can be used as an endpoint in a clinical trial. The idea behind that is that imaging biomarkers are actually everywhere already in most oncology trials. If you look at a recent paper that came out last year on how many cancer drug approvals were based on surrogate endpoints, there were actually 130 molecules that were approved on those surrogate endpoints. And when you looked at what those surrogate endpoints were, they were mostly either response rate or progression-free survival. And actually, these are endpoints that are determined from imaging. So as I was saying, these imaging biomarkers already exist. What we're going to be discussing here is whether we can go one step further. So let's see what imaging, what role imaging can have in these clinical trials. Sorry, I seem to be stuck. I'm going to turn off my webcam just in case that's the problem. So the first possible use of an imaging biomarker is patient selection. So here I'm giving you an example of cervical cancer. It's a uterine cancer, and you might use MRI for treatment allocation. For example, you have patients who have small lesions that would undergo surgery and not be included because your clinical trial wants to consider patients that have chemoradiation. And in this case, we would select the patients on the imaging and include only the ones that have a big enough tumor. You can also use it for patient stratification. Just to keep on that same idea, we would have patients that would all be candidates for chemoradiation, but you would want to differentiate those that have a locally advanced tumor, such as the top patient here, where we have a cervical cancer that invades the parameter, or patients who have lymphatic dissemination, for example, like the patient underneath. So as you can see, you could differentiate good or bad prognosis or other questions and stratify your patients. The last possibility is to use imaging as an indicator of treatment response. And here, just to show you a different type of example, you're seeing CT scans, 2 different patients. The patient up top has a large lymph node here, and you see that after treatment, the lymph node decreases, which puts this patient, classifies this patient as a responder. Whereas this patient down here has a lung metastases, one very big one and smaller ones. And after treatment, well, you see that there are a lot more lesions. So obviously this patient would be a non-responder. So as you can see, imaging can be used a certain number of ways. But we can also think about where you want to put the imaging in the drug development pathway. When you're at a preclinical or a phase 1 study, then you can use experimental biomarkers. I'll give you 2 or 3 examples that will help you comprehend or demonstrate the mechanism of the molecule. When you're going further on in phase 1 or phase 2, then you're really going to be in the demonstration of efficacy or selection of patients or dose adjustments. And then, of course, once you're on phase 3 trials, you're going to have to use a validated biomarker that is accepted by the regulatory agencies. So it has to be a recognized criteria. So you see, there are different roles that imaging biomarkers can play, but you're also going to consider different biomarkers according to what question you want them to answer. So let's just start with the simple ones, the validated surrogate biomarkers. As we said, imaging is often or practically always present in oncology trials, but also in other types of trials, such as cardiology. But here, of course, we're talking about cancer. Why is imaging valued? Well, because it allows objective evaluations and traceability. The image is there, so it doesn't rely on a reported outcome. It's something that can be independently reviewed. But in this case, it is often very basic. Presence or absence of lymph node, for example, or number of lesions or size. Of course, you can think the basic is not good, but there are a lot of advantages to having very simple biomarkers, which are that they are usually robust, that they are very largely available, that they're cost effective, and that they're also intuitive. In oncology, we usually use the size of lesions to guide the patient selection or treatment response. And in a way, it's very easy to understand that something that grows, the tumor is not responding, and if something decreases in size, then it's a good thing. There is a little functional imaging that's recognized as a surrogate biomarker, for example, PET in lymphoma. I just want to say that these basic biomarkers are very sturdy because this is a study that shows different survival curves in metastatic renal cell carcinoma that is treated by anti-angiogenic drugs, which have the reputation not to decrease the lesions in size. But what you see is that when the lesion size decreases, these are all the different populations, well, the more actually I should go the other way. The more the lesion size decreases, the better the survival. So size, even though it is very modest, it's very simple, it does work quite well as a surrogate endpoint. The next slide is just to show an example of PET in lymphoma. As I mentioned, here you have a lot of adenopathies in the chest, and you see that they have a very high metabolism. And after treatment, even though the size practically does not go down, well, the metabolism goes down completely, and this is a sign of complete response. So once again, simple biomarkers, but very sturdy. And so we understand why we use them. But what we think is that we can go a lot further with imaging. And when you're going to be doing preclinical or phase 1 trials, or when you're going to be doing proof of concept, or you're going to want to demonstrate mechanism of a molecule, or make this go, no go decision, then there's definitely more imaging that you can do. This is a representation of the schematic that you probably know, which comes from a publication in Cell, and which highlights all the differences between a cancer cell and a normal cell, and all the mechanisms that are either upscaled or downscaled in these cells. And these are the imaging techniques that allow interrogation of these mechanisms. For example, you can do diffusion-weighted MRI, which is sensitive to cell density or edema or extracellular space. You can do FDG PET, as I said, which is glucose metabolism. You can do perfusion imaging that will interrogate the number of vessels, for example, in your tumor. So all these innovative imaging can allow you to have more information than just size. But we are also moving from hypothesis-driven to data-driven research, and a lot of us imagers are working on what we call radiomics. And in radiomics, what you do is you try to pull out a lot more parameters out of the image of your tumor, parameters that reflect shape, parameters that reflect the type of signal that you have inside the tumor, or what we call the texture, which is very sensitive to heterogeneity. And we know that heterogeneity is an important factor in cancer. So finally, what are the challenges for these innovative imaging biomarkers? Why are we not using them in a routine way today? Well, because they still have to be validated, and there are different steps to this validation. I'm not going to go into it, but it does have to be done. Sometimes those imaging technologies I was showing you are complex to implement. There could be safety issues related to new imaging contrast agents. There are issues of standardization of the image acquisition across platforms. And there are also often several post-processing options, and people don't always use the same. So we definitely have work on standardization to do there. But what often happens is that centers that will be included in the clinical trials are included for their clinical expertise and not their imaging expertise. And sometimes this does not allow to do very complex imaging. So how can we use imaging in clinical trials? And well, just to summarize what I've just said, there are kind of 3 different circumstances. There are the downstream trials where you're after the go, no go decision, where you're going to be using a robust and very widely available imaging technique. It has to be a validated imaging biomarker, and it will be implemented in a large number of centers that can have a very variable experience or expertise. Then at the other end of the spectrum, when you're going to be making that go, no go decision, where you can use more innovative imaging techniques, and Yan Liu is going to talk to you about this. This will allow an exploration or of new imaging biomarkers that will reflect the mechanism of the drug, but this will have to be done usually in a small number of very expert centers. And then you can kind of combine these 2 strategies. You can have what we call companion studies, where we're going to have a large study, but then in a subgroup of expert centers, you're going to have optional advanced imaging techniques. And this allows you to have sort of the best of both worlds, to still have an innovative imaging biomarker, but to make sure that you're validating it on something that's very sturdy. So I'm going to let the next speaker speak, and I'm very happy to introduce Dr. Ravi Karra. So he's currently Senior Medical Director Oncology at Labcorp Drug Development, which is based in Germany. Ravi is a physician with more than 20 years of experience, both in patient care, clinical research, product launch, product life cycle management, from the clinic to the pharmaceutical industry. He has significantly contributed to several ANDA trials, including life cycle management in solid tumors. And he worked as a global medical lead as well as a global medical monitor. He also has extensive clinical development experience in solid tumors, including melanoma, head and neck, thyroid, glioblastoma across all phases and with multiple targeted agents and immunotherapies. So Ravi, the floor is yours.
Thanks, Laure, for the kind introduction. Good afternoon, everyone, and welcome to this session on the challenges in oncology drug development. Well, we have two sections to cover. The first one will be on the current status, and the second one will be on the future needs and perspectives. Let us take a detailed look on where we stand as of now. There has been immense progress made in the past decades. The progress in the immediate past 10 years have been phenomenal for patients. If we take the case of melanoma, 10 years ago, the 5-year overall survival for patients with metastatic melanoma was less than 10%. Now it is close to 50%, mainly due to immunotherapies, notably checkpoint inhibitors, which have dramatically altered the lives of patients for better. The not so good news is that the model for melanoma has not been replicated for other tumors like pancreas, where the difference between overall survival 10 years ago and now is not very significant. We will not get into the details of the why, but if we focus on the what, it is clear that the unmet needs in some types of cancer are daunting, to say the least. To sum up, there has been an immense progress for patients, but unfortunately not for all patients. Let's take a look at the number of FDA approvals of oncology drugs across all indications for the period between 2008 to 2016. There is a systematic and heartening increase in the number of approvals. If we dig deeper, we discern that priority reviews have been increasing year on year. Now, priority review, as we know, is reserved for drugs which, if approved, would be significant improvement in the safety or effectiveness of the treatment, diagnosis, or prevention of serious conditions in comparison to standard of care. This means that between 2008 and 2016, the drugs approved by FDA met one of the aforementioned criteria, which can be generally translated into more effective and less toxic treatment for patients. If you look closer, we find that a lot of progress has been in the area of immunotherapies, whether it be passive or active. This field has shown that a lot of activity can indeed mean progress. Currently, there are close to 5,000 products in various stages of development, including T-cell targeting modulators, cell therapies, etc. and etc. And I am confident that this trend will continue to grow for the betterment of patient lives. Despite the science getting ahead by leaps and bounds, the methodology of testing and validating the science has not caught up with the progress, meaning the development of drugs is still mostly linear and sequential, with the traditional preclinical followed by clinical development steps of phase 1 to phase 3, with the increase in the size of the trials and the duration of the treatment associated with preset regulatory milestones leading to an approval. Now, this worked in the era of chemotherapy, where drug development was mostly serendipity. Why is this model unsuitable for the immunotherapy era? Several reasons. Number one, because of the long development timelines. Access to promising drugs takes time for deserving patients. The second reason, the longer the duration, heavier the financial investment. And the third and the most important reason, along the development pathway, patients are exposed to drugs that may not benefit them, and in the worst case scenario may end up harming them. This linear and sequential model is ill-fit for the era of immunotherapy. Let us take a look at the future needs and a few perspectives. Now, what would match the progress in science with the development would be the development that feeds from the science and feeds back into the science, meaning we need to have a continuous and integrated development where the learning of today is not waiting to be implemented tomorrow or the day after, but the learning from today is integrated and is put into use today. So this means that we need to have a process where the learning is continuous and preferably starts early. What do I mean by that? I mean that we need to have a proof of mechanism or a proof of efficacy as soon as possible. This would entail that we design and develop trials that are flexible, continuous, and integrated, and can generate data faster, and based on which the trial generating that data can be modified, or new trials can be set up based on the data. What this kind of development would lead is to hopefully an earlier readout for efficacy and maybe even an inkling into the subgroup of patients that are benefiting or may not be benefiting. If this is combined with possible robust biomarkers at each step of this integrated development, of course, the biomarkers correlating with the pharmacokinetic and the dynamics of the drugs, then we may have sufficient data that could actually withstand not just the scrutiny of the regulators, but also that of the payers. Now, is this a flight of fantasy? I do not believe so. Why? If you look at the way pembrolizumab was developed, this was by no means linear and sequential, but was integrated and continuous, with one trial feeding into another and the learning being integrated into sometimes the same or a different trial. The end result was that pembrolizumab was available to patients in a period that was much shorter in comparison to the general timelines for an oncology drug availability. Now, is this a replicable model? I think yes, if we have the key data that can answer the following questions. Do we know the right target and the right patient? Do we know the right dose and the right time? And we do have the right safety and the right efficacy. Let us delve deeper into each of this. If you look at the right target, how can we assess target engagement efficiently? Would we be able to have a rapid assessment of target engagement? For example, could we use imaging? Could we have an idea on the indication on off-target effects, on adverse effects? How can we translate the preclinical learning into the clinical space? And if you look at the right patients, do we have the data on which patients are likely to benefit? Can we get more insights into how the microenvironment is going to interact? Can we start with small studies to learn with? And how does the correlation of the biology reconcile with the clinical outcomes? If we move on to the right dose at the right time, for the right dose, can we robustly base the dose on the rapid assessment of the starting dose in animal models to humans? And if we dose escalation, should it be targeted or should it be just like what we did prior in chemotherapy trials? And should the dose be based on target versus off target effects? And to assess the right time, how do we do that? Should patients be treated as a part of a first line treatment, or in the late stage of their disease. Should patients be treated with monotherapy or with the combination treatments? Now, if you look at the efficacy and the safety, can we make sure, for example, for the safety part, how early could we have an assessment and how long could we follow up the patients with a seamless design? And can we sometimes, the learning from the clinic, back translate that into the preclinical space? And similarly for efficacy, does tumor biology give us an indication of which patients might benefit or not? And targeted small studies, could functional imaging help us, or the correlation of the biology to outcomes? Well, those seem to be an endless stream of questions. Now, let us try to seek some answers. Well, we know that imaging has and will continue to help us answer some of the questions I have put forth. Traditionally, we have used imaging for diagnosis and to assess efficacy via tumor measurements. With the advent of targeted imaging, it is possible, and the question would be, is it possible to do more, to obtain more and specific information from imaging? Yes, we can, says our next speaker. It is my pleasure to introduce Dr. Yan Liu, the Chief Medical Officer of Median Technologies. Yan Liu is an imaging expert with around 20 years in clinical and academic research experience, with the past decade dedicated to the integration of imaging in multi-center oncology trials and the validation of imaging biomarkers. Previously, Yan served as the head of the Translational Research, Radiotherapy and Imaging Department at the prestigious European Organization for Research and Treatment of Cancer in Brussels. She has published multiple book chapters and several peer-reviewed papers in high-impact journals, and was also a member of the RESIST working group. Yan, the floor is yours.
Thank you, Ravi, for a very kind introduction. And also your excellent talk provide us the insight on the challenges of drug development and the importance of the precision medicine, and to provide the right drug to the right patient. So now I will give you some idea on how imaging solutions can be used for clinical trials. So we know that imaging plays a key role in oncological trials, and some of them we're very familiar with. For example, using the standard efficacy evaluations based on the criteria to support the study key endpoints. Some of them, they can based on size, volume, such as RECIST, RANO, mRANO. Some of them, they are more based on metabolic assessment, such as PERSIST or Lugano. But as a plus, we also provide solutions for biomarker discovery. Some of them, we can extract those information from the lesion, from the tumor. Others, it could be more host-driven biomarkers, such as the analysis on the lung emphysema or the index for the sarcopenia, or we can see the liver or the lung fibrosis status. All this aim to provide more insights into tumor and also the host microenvironment based on quantitative imaging biomarkers. And also, as Ravi and Laure just explained, imaging biomarkers can be implemented in many steps of clinical trials to either confirm the eligibility, to verify the target engagement, to assess early response, monitoring safety, surrogate endpoint, etc. And all that can help the drug developers to understand better the mechanism of action and also go deep insights on the subcohort, and eventually to make appropriate go, no-go decisions in an appropriate way. However, before we use them, we should not ignore the risks of the implementation of those novel imaging biomarkers. And now I will share with you some examples of the risks. So one of the examples of the risk is the risk of variability. The variability is the one that actually the biggest challenge faced during imaging-based trials. It can happen when we have the imaging capture or when we do the imaging interpretation. So the imaging acquisition and the variability cost can be linked to different scanner, different contrast media, different measurement parameters, patient position, for example. And that a standardized imaging guideline will be helpful and to control those variabilities. We can also have variabilities related to our read interpretation. We can see a single reader that we may have the intra-reader variability. So for example, the interpretation difference, and that can be due to the stability of a reader and also due to difficulty of certain case. We may also have inter-reader variabilities. That's the interpretation difference among multiple readers, and that's usually linked to the reader scale. We may have variability related to the reader bias. That's a reader knowledge of a treatment condition influence the interpretation of the image, or related to reader fatigue, because we give too much scans to one reader and eventually that decreased capacity to have a correct interpretation. I will say that certain variabilities can be controlled, and also, for example, the bias can be controlled by certain methodology, and the reader fatigue can be avoided if we can balance the workload for our readers. Some variability, as mentioned, that can be controlled thanks to technology. The system can present images to readers and provide solutions with automatic lesion identification, quantification, tracking through all imaging time points. So here you can see the system can segment the lymph nodules and have the 3D volume. And also it works in the liver nodules. You can see here that the system can automatically segment the lesion that can reduce the variability. And of course, reader can also correct it if he believes it is not accurate enough. So those tools are helping to decrease the variability. Another example I would like to show you is a risk that's related to the poor quality. And it's extremely important when we implement functional molecular imagings, because they are highly dependent on imaging quality for those quantitative functional biomarkers. This is a study which we may use as a lesson learned. This is a sub-study from Publique trial and published in 2015. The aim of the study is to test the hypoxia that patient with FDG PET response, so decrease more than 25% SUV max, that the PFS is 12 weeks longer than the patient without PET response. So use the metabolic images to assess early response. So in this sarcoma trial, total 44 patients were enrolled in the sub-study, and 35 patients among the 44 with good visual quality. However, when it went to details about the quality metrics, it shows very low compliance to the imaging acquisition guideline. As we know that the uptake time is very important parameter from the PET imaging quality check. So in this paper, the authors compare the uptake time come from the imaging transmit forms and the DICOM header. And also they found that the data noted in the DICOM header have much less compliance compared with imaging transmit forms. And usually DICOM header is automatically calculated by the system, while the imaging transmit form is filled by human. So the DICOM header are more reliable. So in this case, based on the DICOM header, only 31% of the cases which complies with imaging acquisition guideline, which requires imaging be performed 60 minutes plus or minus 5 minutes after injection. Well, when we check the intervals between the baseline and the follow-up, and we also find that only 66% of the scanners are within 10 minutes interval. In addition to that, there was also artifact. So as a result, less than half of the patient could be used for the quantitative analysis, and no conclusion can be drawn due to the inadequate sample size. Therefore, it is essential to identify the risk and set up appropriate risk management for imaging-driven trials. So as an imaging CRO, we work closely with the sponsor to mitigate such risks at its trial initiation, that we can provide the right rationales to develop a protocol, and also to identify those potential risks. We then also provide the support for site qualification, for machine collaboration, and also for the standardization. During the trial, we can also conduct a central reading with robust methods, and eventually that we can provide reliable data to the sponsor. So we know that those kind of imaging implementation could be complex, but however, with robust imaging modalities and also with all the steps behind, we can keep such kind of complex design also as a successful story. So here is a story that I want to share. As we know that targeted agents are often investigated in unselected end-stage cancer patients, make it difficult to fully evaluate its activity and also impairing translational research. So one way to resolve this issue is to perform a window study where a targeted agent is given between the diagnosis and the surgery. So in this ERTC 9111, 30 patients were enrolled with 5:1 design, and the primary endpoint is to use FDG PET to assess the drug efficacy. And also, it included multi-parameter MRI, including DCE, so dynamic contrast enhancement, and also diffusion-weighted imaging as a secondary endpoint. So those images have been performed at day 14 to assess early response. And based on the previous experience on the sarcoma trial, robust QC steps have been implemented for the site qualification with imaging guidelines, the front-end collaborations, etc., and also the real-time quality control and a central reading were performed by independent readers. So in this proof of concept trial, it shows that it is feasible to conduct multinational challenge window studies with modern imaging modalities. And also the results also demonstrate a very high rate of FDG PET response compared to the RECIST 1.1. So all this information give a go signal and provide high values for the next step trial design. Therefore, in summary, at the trial initiation, it is critical to have right protocol design based on scientific rationales, to also select the appropriate criteria, imaging modalities, using the right imaging window to detect the drug effect, and also to have a powered study which can help us to draw a conclusion. Before the site activation, we should also provide imaging charter, define workflow, and also provide the trainings to site. That will also guarantee the good quality later on. During the trial, we can also continue to do the prospective imaging QC and to provide in-time feedback to site about the quality. And also, we perform the central reading and we monitor our reader performance in a regular manner. And finally, we can make decisions based on good and reliable quality data. So thank you a lot for your attention. Now I will pass that to Laure to draw us the take-home message. Thank you.
Thank you very much, Jan. I'm just having trouble putting the slide up. So I hope we convinced you. It's going back and forth. I don't know. Sorry about that. I hope we've convinced you that imaging can bring a lot more information than what we are currently using. But I think that one very important point that particularly Jan raised in the last talk was that it's important to include the imagers early on at this, really when you're at the stage of the trial design, because if you want to exploit all the information that imaging is giving you, you have to include it upstream and not afterwards thinking, oh, we have images, maybe we should have been looking at them or pulling something out from them. And if you want to include imagers early, that means, of course, that you have to have a budget for this imaging, because that is sometimes forgotten as well. We tend to think that we have routine clinical imaging and that we're going to be able to use that, but I really think that if you want to pull out the most information, then you have to invest on imaging, just like you invest on biological biomarkers and all the other data that you collect for your clinical trial. So those are the little take-home messages. I really want to thank Ravi and Jan for these great talks. And I think we'll just open the round table now. If you have any questions, you can post them on in the little panel that you have. We would be really happy to answer them. Let me give you maybe 1 or 2 seconds to put in your questions. And otherwise, I don't know, maybe we have questions for each other. So I'm not seeing any questions from the audience for the moment. Jan, I think when we were preparing this, we were having a discussion about diffusion-weighted imaging, because I know that a lot of people, I know that you have experience with diffusion-weighted imaging. You actually mentioned it in one of your slides, but you were talking about PET. Were there any results from diffusion imaging in that study?
Oh,
sorry, that's
yes,
a bit of a
yes.
trick question.
Yes, but the diffusion-weighted imaging results have not been published because the PET was the primary endpoint. And also, that's your very interesting insights from that study, because we do see that there was a decrease of certain patients, and also there was non-responders. And we also see the ADC values changes from certain patients. But however, for that study, due to the standardization for diffusion-weighted image, and we also have quite limited data really to draw robust conclusions compared to the PET, because the PET message is very, very clear, and also it's quite robust. And this is the reason that it was not shared with the other community. But however, I will not hide the difficulties when we implement the diffusion-weighted imaging and DCE that time, because we also talk about the phantom. And you know that with the phantom, we have ice phantom, and we need to also move the phantom from side to side to do the machine calibration. So actually we did that. And I think all this experience help us to design the next study when we want to implement diffusion-weighted imaging driven study. And we can also use those information to have better, more powered studies for the next one, because in this case, we have limited numbers that the data can be really interpretable with the diffusion-weighted imaging.
Thanks a lot. So there's a question from the audience. I guess it would be for Ravi and or Jan. So the question is from Susan Burak. How do you deal with different regulations with respect to imaging in international clinical trials? So I don't know, Ravi, if you want to.
Yeah, sure. So, well, that's a great question from the audience. Well, we always see that, leave alone the international, we have seen even at the national level, even between different sites. For example, Jan gave that study on the successful case for the EORTC 90111. In fact, both Jan and I worked on that study. We were working with the top-notch sites with top-notch images, and still we had an uphill task on the harmonization of not just the acquisition of the images, but also the reading and the interpretation, so on and so forth. Given that, I think it's uphill task to talk about harmonization at a global level, but what we do and what we have seen and what we have achieved multiple protocols is when we look at pivotal trials where, for example, we have a central review of images, and then there is a certain standard protocol for how the images must be acquired and how they must be sent, how they must be read, how they must be analyzed, and so on. So this has been happening for quite some time now. On the other hand, out of clinical trials, I think it's a bit complex to make sure that the images, the imaging and the technologies align. We see this, for example, PSMA CT, PET CT for prostate cancer. Well, if you want to make this as a standard, some of the investigators love it, and it's a very efficient method. But on the other hand, how many of the sites have the technology? How many of the sites can afford the time and the money? And are the site personnel trained? And how is the tracer availability? So if you look at all of these factors, what it means is that some of the things can be done at the top of the pyramid. Now, if that imaging and the technology has to percolate down, it's a conscientious effort, not just from the medical community, but it also depends on the healthcare ecosystem. For example, are the payers willing to reimburse this high-end imaging? So that becomes the key topic. So that's a good question, but the answer would be, in short, yes, it's possible, at least in a few select countries. For example, we can start it in Western Europe, but it's a question that needs to be solved not just within, as I mentioned, from the healthcare community, but the healthcare ecosystem as a whole.
Yeah, definitely. Thanks, Ravi. I may have something to add. Is that from regulatory part, they will need to have qualified imaging biomarkers, and that's really need to be based on evidence. So the evidence can be different results from multi-centers that we generate from retrospective studies, and then we can also see that whether we can have a cutoff, a predefined threshold to say responder and non-responder, and with that, ideally, is we can have a prospective trial to validate such kind of cutoff. I think now these days, the biggest challenge is the standardization, but we can provide an standard acquisition protocol. Then is the further clinical validation. So how we can rely on those biomarkers, and that also will need a resource to validate and qualify those biomarkers. And I also personally believe that this will be the efforts not only come from academia, but also from private industries. So this kind of biomarker qualification can really put us a reliable biomarker for drug development. So yeah, so this is some of the idea from my side. Thank you.
Yeah, and thanks a lot. I just wanted to add something to that. I think one of the reasons that we have the feeling that imaging is not very well standardized is also because imagers have historically not been involved much in clinical trials. I think we always assume that it's just going to roll, right ahead and work because everything is standard and everything is clinical routine. But yeah, it's not true. And I think that we work a lot on standardizing biological markers. We work a lot on standardizing the clinical questions and how you fill in the ECRF, etc. And we haven't spent enough time worrying about the imaging. And once again, this is kind of historical, and imagers are learning to participate in clinical trials, but I think it's important for the promoter, the sponsor, to actively include them, because it's only by participating in clinical trials that imagers will get better at it. So I definitely see a gigantic difference today in imaging departments compared to 10 years ago and even 5 years ago, because there's an awareness, I think, of the importance of imaging in the imaging departments, and a desire of being involved. But once again, it has to come from both sides, I think, and the sponsor has to go, interrogate the imagers and make sure that they know what they're doing, and as I was saying, I mean, I don't want to say that we're all worried about money, but include that in the budget. It's something that you have to plan on ahead of time. So we actually have another. Yeah, we actually have another question from Eva Ristova. In how many of the clinical trials? The question is kind of, I can only say part of it, but is there our harmonization of the equipment and not only the protocol? So I hope I'm getting this question right.
Yes, thanks for the question. Very, very good one. As we mentioned also before that when we have the qualification of the sites, that's also including the qualification of the scanner, especially when we do quantitative image biomarkers. That's extremely important. I think from the European side, so it's a spin-off of their E&M. They did a very, very good job, and they also provided the support for clinical trials which quantitative imaging will be implemented. So they also provided a service to qualify those kind of scanners by providing the phantom, providing also the methodology for the analysis. So those are the efforts that from the European side has made. So scanner qualification is very important, because only based on that we can rely on those data and we can pull those data together for this comparison. Thank you.
Ravi, do you have something to add, or?
No, I think I would go with what Jan said. I have nothing in addition to that.
Yeah, I guess maybe what I could say is that it's important to work on harmonizing the equipment when it's necessary, because I also find that sometimes a lot of things are asked from us from the imaging department point of view that is a lot of extra work and that doesn't contribute so much. So I'm just thinking of something as simple as CT imaging for evaluation of size of tumors. Then honestly, I'm not sure there's much qualification of equipment to put in there. And then when you ask that, it's just extra work and it's kind of, yeah, not efficient, right? So I think it's important to have imaging experts. Once again, I'm sorry I'm insisting, but I think it's important that really guide what you need, when you need to harmonize the equipment, when you need to harmonize the protocol, so that you really make it worth the effort. We have another question. That's great. Our audience is really out there. So Earl does PET CT harmonization, so we're on the same subject. But what about functional MRI harmonization? Is this being done in clinical trials? Ooh, difficult question.
Indeed. Because the functional MRI, and also it's also because MRI have a lot of sequence. I think Laure, you're an expert on that. I just start and then you can add what I say. So there was many multiparametric MRI behind, and each of the technology we can do standardization. But as you asked me the question about the diffusion study that I mentioned before, there was some also logistic concerns behind, because of the collaboration, the resource, the timing, the data analysis. If it not passed, how you will do. So all that, I think, are the risks that we should identify at the beginning. And if we prepare early, I believe that we can solve those problems. But absolutely, that additional resource that we should consider and put in place before we start the trial.
Yeah. Sorry, Ravi, go ahead.
No, just to add on, I fully concur with what Jan said, and Laure, I go back to what you said. Involve images right from the start, because what I have seen is time and again, if you really want to get a good quality imaging data set, I always insisted that the images be a part of the team and be a sub-investigator right from when you are reviewing the protocol to make sure you want to join or not at a later stage, but right from the start, so that they feel a part of the team. And in fact, they should be a part of the team, because based upon whatever drug is getting approved as of now, what is it based on? It's all on imaging. So when you have that kind of a collaboration, things are going to go smoothly. But on the other hand, we also have to realize that they are stretched. They have to spend so much time on clinical trials, and also they have their daily routine of patient care, daily routine of administrative tasks, teaching, so on and so forth. So it may not be replicable. Once again, the top-down model, the percolation could be a bit difficult, but all said and done, I think, as you rightly said, we have seen gigantic improvements and strides in the right direction, and I can only concur that this is going to move forward.
Yes, and I think we have good examples. For example, in brain imaging, there is a standardization of functional imaging protocols in MRI that's led by URTC, among others. In prostate cancer, it's very standardized the way you do a prostate cancer like the diagnostic MRI. So I think we're really going forward. But just to kind of see the other side of the coin, I think it's important also to develop biomarkers that are kind of robust to the variation of equipment. So it's also our job as imagers to find those biomarkers that do function in a large set of situations. But once again, as both Jan and Ravi said, it also depends on the question. When you're going to be at the very early stage, then you're going to have a small number of centers, maybe only one center, and then you can really push the imaging very far and have something extremely extremely expert and precise, where you're going to be doing a lot of precise quantification. And then when you're going to want to widen it to others, and that is part of the development of the biomarkers, you're going to want to work on its robustness and how it can be implemented in a more diverse setting of equipment.
Definitely. And Laure, you're an expert managing also big data. I think, as you mentioned, something will work. It is something practical, can be implemented easily, and people will use that. So how do you think about the AI in the future for the drug development from the imaging side? If you could give us some insights on that.
Well, thank you. Well, I think AI is a tool to extract information from images. So to me, it's not, I mean, we're using different methods to discover the biomarkers, but in the end, the goal is the same. It's getting a biomarker that works and that relates to the question that you want. So it's mechanism, it's efficacy. It can be toxicity. We haven't talked about that much, but imaging is interesting for toxicity as well. So to me, it's just another way of discovering biomarkers, and it's a very interesting way because you can use a lot of retrospective data, and you can find biomarkers that haven't been, imagined yet. But once again, in the end, you're still going to have to use a biomarker that you've validated. So yeah, it's just the discovery phase of the biomarker that's different. But I'm definitely optimistic. At least that's what I'm working on.
Yes, I think well, we are at the top of the hour. We have 2 minutes more. Thanks to both of you, the speakers. I think we had a very great session, and I would also like to thank the audience members. I think we had a good number of audience join us, and we had some interactive questions as well. So I thank all of you and also the organizers. Thanks to everyone once again, and have a nice rest of the day.
Bye-bye.
Thank
Have
you.
a nice day.
Bye.